so today, right now I'm going to talk about chapter 13, which is meiosis and sexual reproduction. And this chapter, thankfully, is a lot shorter than chapter 12, which was mitosis and the cell cycle. It's a lot shorter because meiosis has a lot of the similar mechanisms that we went over in mitosis. So with mitosis, it was cell division. Meiosis is also cell division. It also has very similar phases of, like in mitosis, it has prophase, metaphase, anaphase, telophase. However, the whole purpose of meiosis is completely different. Okay. Remember, mitosis was for the purpose of producing more of the same cells for repair, for growth, for development. But for meiosis, the whole purpose of meiosis is to produce gametes, or sex cells like sperm and egg for the purpose of sexual reproduction. So there are animals that are going to reproduce sexually, but in order to do so, they have to produce gametes, which are sex cells like sperm and egg, in a process called meiosis. So that's what chapter 13 is all about. It's the process of meiosis. And this chapter will definitely be a lot shorter than chapter 12. All right, let me go ahead and play from the start here. Okay, so like I said, animals that reproduce sexually, they're going to produce gametes via the process called meiosis. And then, there are organisms that reproduce asexually, like this amoeba, which my face is blocking, but you've seen it before in chapter 12 PowerPoint. The amoeba is a single-celled eukaryotic cell. It's eukaryotic, so it has a nucleus, it has all the organelles, but it's going to divide and reproduce asexually, producing clones of itself. So the amoeba will just grow really big until it hits a certain big size, and it's going to divide in half and form two amoeba. So amoebas, protists, paramecia, those are examples. Protists, paramecia, amoebas, those are eukaryotic cells that can reproduce asexually, as can yeast or fungi. There are some simple multicellular organisms that can also reproduce asexually. Like in this case, this is a hydra. Hydras are related to jellyfish, because they also have stinging tentacles that can paralyze their prey. And you can see here, this is the mouth, and that's where the stinging tentacles will paralyze their prey, and that's their mouth, and then their prey would be digested in this gastrovascular cavity. Here is a little baby hydra that's budding off, and it can reproduce asexually that way. Okay? I apologize if you can hear my son in the background. I feel like I'm lagging again, but I'm just going to keep on going. So my son, he's playing Pictionary, and so he's like screaming in the background because he's trying to guess what they're drawing. All right. So this is this slide is just showing you how not all organisms reproduce sexually. They do not do meiosis. They just simply clone themselves by mitosis. So I want you guys to think about a few things, and let me just lower the volume because I forgot Fillmore is going to quack here. There he goes. Maybe you guys want the volume up, huh? Okay. What are the disadvantages of asexual reproduction? What are the advantages of asexual reproduction? So let's talk first about disadvantage. One of the disadvantages of reproducing asexually is that you're just producing clones of, they're just producing clones of themselves. So, let's say that there's some kind of mutation that is not a benefit to that species, and that mutation just gets passed down, then they can potentially, go extinct. Or let's say that there is a pathogen, maybe a fungus or a virus or a bacteria that can infect one member of the population. It can spread and kill the entire population. If they are all clones of each other, then they're at a disadvantage, okay? But what are the advantages of reproducing asexually? So what's the advantage of being able to just bud off and produce another hydra? What's the advantage of just splitting by mitosis and producing another amoeba? Well, first of all, in the animal kingdom, sexual reproduction is a big deal, right? You have You have to go find a mate, and then you have to attract the mate. It's just a lot of energy involved with finding a mate and reproducing sexually. So the advantages of reproducing asexually is that you don't have to devote a lot of time and energy into finding a mate or even just producing gametes. So the time and energy is not spent in meiosis or in finding a mate and reproducing sexually. Okay? Alrighty. So asexual reproduction by mitosis produces identical daughter cells or clones. Like here's Fillmore, he's cloned himself multiple times over, same number of chromosomes, same genetic information, and the disadvantage of that is if there was some kind of virus or pathogen that could kill one Fillmore, then they could all, die from the pathogen, and sadly Fillmore would be no more. Oh, wow, I just made that up right now. Okay, how about sexual reproduction? It occurs in complex multicellular organisms, and we're producing egg and sperm that are going to join up to produce a little zygote. So how do we make egg and sperm? I mean, do we make egg and sperm by mitosis? No, we do not. Because what if we did? And it would be like this. If we did produce sperm and egg by mitosis, then our egg would have 46 chromosomes. Our sperm would also have 46 chromosomes, because remember the starting cell has 46 chromosomes. Then if we produce sperm and egg by mitosis, the daughter cells or the sperm would also have 46 chromosomes, and then you're going to end up with a zygote at fertilization. It's going to have 92 chromosomes, and that number is not going to work for human development. 92 chromosomes is too many. That does not work. Okay? So we do not produce sperm and egg by mitosis, because that whole process of mitosis is producing exact same daughter cells, 46 chromosomes to start with, and then you end up with 2 new cells with exactly the same number of 46. And so mitosis is not the way egg and sperm are produced. Here is a picture of someone's chromosomes. Okay, this is an actual microscopic photo of someone's chromosomes actually this is a picture of G1 phase, where they cause the DNA to coil up really tightly to form chromosomes. So in G1, you wouldn't see them in chromosome form, but in the laboratory, you can apply some kind of chemical where the cell is going to condense its chromosomes in the G1 phase. Okay, so that's what's happening here. Okay, this is what we call a karyotype. Okay, chromosome number 5 is kind of blocked because of my head. But let's take a look at the other chromosomes. Let me grab my annotation tool pen. Come on. Okay, so we have, if you count all of these chromosomes, there's 46 or 23 pairs. Okay, so we have 46 chromosomes or 23 pairs. Why are they in pairs? Well, you got 23 from mom and 23 from dad. Okay, so here's pair number 1. Chromosome number 1, it looks a little bit shorter, but in actuality, there's some kind of folding here. Remember, it was a picture. They took a picture of someone's chromosome. So at that point in time when they took a picture, there was probably some kind of folding, but the chromosomes actually look exactly the same. Let me give you guys an obvious example. Take a look at chromosome number 9. So let's say this one's from your mom. Let's say this one's from your dad. So each pair here, one is from mom, one's from dad. See the banding pattern? Do you guys see those like dark lines on them? Okay, so chromosome number 9 has a specific appearance. Every time I look at my picture, I'm like lagging. It's weird. I mean, look at chromosome number 7. It has a specific type of banding pattern. Okay, there are genes on these chromosomes, like gene for, let's just pretend, I don't know if this is true, but on chromosome number 7, let's say that from your mom, you got the gene for brown eyes. And then from your dad, let's say that you got the gene for green eyes. Okay, so what that means is that you guys have 2 of every chromosome, 1 from mom, 1 from dad. You have 2 of every gene, 1 from mom, 1 from dad. And all of those genes are found on these chromosomes. Okay, so this is what we call a karyotype. I should have written that out here. A karyotype is basically a photo of someone's chromosomes where they've arranged the chromosomes according to size. They've been paired according to size, chromosome number, and they can tell that this is chromosome number 7. This is chromosome number 14 because 14 is smaller than 7, and then you can also tell by the banding pattern because it's kind of, I mean, when I look at number 11 and 12, don't they look pretty much the same size? But they can tell just from the banding pattern which chromosome is which, okay? And we can also tell that this is a female because there are 2 X chromosomes. And because there are 2 X chromosomes, that's going to mean female. Okay, so the sex is, biological sex is going to be female. So to say, what I want to say here is that everyone, we have 2 of every chromosome. They're in pairs, and specifically we call these pairs homologous pairs. I'm running out of space, but I'm just going to write it here. Homologous pairs. And I'm going to talk about what that word means later, but we have 23 pairs, homologous pairs. Okay? Oh, and I did want to say too that the first 22 pairs are called the autosomes. Every time I say autosomes, I want to sing that AutoZone song, okay? But autosomes. And then the last pair is the sex chromosome. So we have 23 homologous pairs. The first 22 are the autosomes. And then the last pair is called the sex chromosomes because they determine the sex of the person, okay? All right, moving on to the next slide. Okay, 46 chromosomes, 23 pairs, and that's the female. All right, take a look at this karyotype now. And we can tell from this karyotype that it is a male, okay? So it's going to be genetically, let me just get my pen. I don't know why my pen disappeared, but it's a genetic male. You can see an X and you can see a Y, okay? So again, you're going to see, and look at this chromosome number 1. See how it's bent? So it was just a snapshot in time. They took a picture of it. It just so happened chromosome number 1 was kind of like, you know, folded, bent. It's like if you take a picture of me, like right now my eyes are closed, you know? So same thing, they took a picture. That's why all chromosomes are not stretched out perfectly because it was just a snapshot in time. But you can see here 23 pairs. The first 22 are called the autosomes, right? Not autozone, autosomes. And then the last pair, the sex chromosomes. But you have 23 pairs. Okay, next. Oh, I did want to point out too that, you know, females, we have 2 X chromosomes, so 2 of the same sex chromosome, whereas males have 2 different sex chromosomes. They have 1 X and 1 Y. And notice how the X chromosome is pretty big. It's pretty big. I mean, it's like almost the same size as like the, you know, the 4th or the 5th chromosome. So females, we have 2 largish chromosomes, whereas males have 1 X and then 1 little tiny Y. The X chromosome has over 200 genes on it, whereas the Y chromosome only has about 50 genes on it, okay? So females, because we don't have that Y, we don't have those 50 genes, and there's this one gene on the Y chromosome called the SRY gene. When that gene is expressed, usually at around the 8th week of development. So when a woman is pregnant with a fetus with the Y chromosome, at the 8th week of development, that gene is actually turned on, it's expressed, it starts to turn that fetus into a male. It's a physical male. It's going to grow testes, etc. Okay? So, you know, like, you know, if you know, the parents want to find out, oh, you know, what kind of baby are we having? What sex baby are we having? You can take photos, right? Sonogram photos, and then if you see the penis, testicles, and you know that it's male. And it's because of the SRY gene being expressed at around the 8th week. Okay. All right. So moving on. Oh, you know what? I just pressed the space key. I forgot about that. So as I mentioned before, we have 2 of every chromosomes, one from mom, one from dad. We call that pair homologous pair, okay? That word homologous, and maybe we can look down here. Homo means same. Logos in Greek means information, okay? Homologous, so same information. So as I was pointing out when we were looking at the female karyotype, remember how I said, like, let's say that maybe your mom gave you the brown-eyed gene and your dad gave you the green-eyed gene. Like on that chromosome, you'll find at the exact same place, location. So maybe I'll just draw it here, okay? So this is maybe from mom, this is from dad. So at that exact location, you're gonna find the gene for eye color. So that's what we're talking about. On the pairs, you're gonna find the same information, the same gene. Well, not the same gene, I should say same trait, eye color. You're gonna have 2 of every 2 genes for every trait. One from mom, one from dad. Okay? Oh, and then I wanted to say, like in that picture of the karyotype, you saw the chromosomes as single strands. But then remember, you guys, the cell cycle, those chromosomes are going to double in number. At what phase? Do you guys remember in the cell cycle, what stage or phase of the cell cycle is where the DNA is being doubled? The DNA is being synthesized. And I hear you guys all saying the S phase, so that's good. So at the S phase, the chromosomes double up and then you end up with double-stranded DNA. So you're going from single-stranded to double-stranded, okay? You know, in the karyotypes that you guys saw earlier, I said that you could see the single strands because what they did was they added a chemical to cause the chromatin to coil up and form chromosomes, okay? No, I'm recording. Can we talk later? All right, sorry, my son's here and I'm recording and I need him to go away. We'll talk later, okay, bud? So how do we make sperm and egg? Yeah, maybe you should really go. Okay, cool. Ask Daddy. Okay?
No, I had to do things.
Please. No.
I was playing it.
Thank you. So in order to make sperm and egg, can you just go, bud? You'll learn this later on. Okay? Thank you. All right. We must reduce 46 chromosomes to 23. Yeah, he'll learn this later on, like in high school, okay? From 46 to 23, you have to reduce the number of chromosomes, right? Because at fertilization, you want to make a zygote with 46 chromosomes, which is the normal number, okay? So what's going to happen is you're going to start off with a cell with 46 chromosomes. It's going to go through meiosis to produce egg or sperm with 23, with half the number of chromosomes. So basically the whole purpose of meiosis is to halve the number, of chromosomes to 23. Because at fertilization, then you'll end up with a zygote with the correct number, 46. So really, that's the whole purpose of meiosis, is to halve the number of chromosomes. Meiosis is the production of gametes. So we said gametes are sex cells, okay? We have to halve the number of chromosomes. So here's some vocabulary that you guys need to know. Diploid, okay? Di means 2. Ploid means sets. Ploid means set. Ploid means set, okay? So diploid means 2 sets. You're gonna have 2 sets of chromosomes. Why do you have 2 sets of chromosomes? One from mom, one from dad. And again, the whole purpose of meiosis is to halve the number of chromosomes. So you're going from 2 sets to haploid. Hap means 1. Okay, so it's haploid means 1 set. Okay, and the symbol for diploid is 2N. Okay, 2N is a symbol for diploid. Haploid symbol is 1N. So you're going from 2N to 1N. You're going from 2 sets to 1 set. Humans, we're going from 46 to 23, okay? I think it's chimpanzees. They have 48 chromosomes, okay? I think if you look at chimpanzees, if they're going to make gametes, how many chromosomes would their egg and sperm have? It's going to be half of 48, which is 24, okay? So depending on the animal that we're talking about, it's the same idea that they have to have the number of chromosomes to make gametes. And fertilization is going to bring those gametes together, restore the diploid number, and then that diploid organism is going to go through mitosis to grow and develop. So do you guys see how meiosis and mitosis are connected? And we're going to go back to that idea later on. Okay, but anyways, here, let's start over here. So we have a woman, we have a male. Meiosis is taking place in the testes of a male. Meiosis takes place in the ovaries of the females. They go through meiosis. There are cells called germ cells. They're called primordial germ cells, but oftentimes they're just referred to as germ cells. There are germ cells in the testes and the ovaries that go through meiosis. Okay, they produce sperm and egg, which happen to be haploid. Okay, and then at fertilization, the haploid numbers come together. N plus N makes 2N. And now you have a diploid zygote, and then it goes through mitosis to develop into a mature, sexually mature person, organism, and then the whole process goes round and round. Okay? Oh, but I should say too that the process of meiosis doesn't happen in the testes until puberty. Okay? For males, it happens at puberty. For females, it happens during fetal development. I know that sounds really weird, but the beginning of meiosis for females and ovaries, it happens during fetal development, and then when we're born, we are born with about 2 million eggs that are still going through meiosis. Okay, they're not fully developed, but they're forming. But then males do not produce sperm until puberty, and they continuously produce sperm until they die. Whereas females, we have a limited amount of eggs. Okay, by the time that a woman is ready to reproduce or sexually mature, she has about, I think it's 200,000 eggs available. Obviously, she's not going to have 200,000 children, because as the days go by, eggs will slowly disintegrate and die out, and then by the time she hits what is it called, menopause, she may only have like 100 left, but they are just they're going to eventually disintegrate within the ovary, and that's what happens to those eggs. They never get released. Okay. Sexual reproduction life cycle, and I kind of already just went over that in the previous slide, right? We make gametes, and that fertilization, you make a zygote that is now diploid, and then that zygote, when it sexually matures, it goes through meiosis to produce gametes, etc. Okay. Oh, okay, yeah. And so one thing that I didn't mention, because I failed to notice here, is you take a look at the zygote here. This is the zygote, and it has a mix of chromosomes from mom and from dad, right? So this is the egg, this is the sperm. So let's just say the red ones are from mom. Let's just be very traditional. Red is for mom, blue is for dad, okay? And then when this organism makes gametes, it has a mix of those chromosomes. Some are from mom and then some are from dad, right? Okay, so what Philwar is pointing out here is that when you make gametes, you're going to be passing on a combination, a mixture of chromosomes from mom and dad. So we're mixing things up. We are mixing it up for the next generation. When you have kids, you don't know which one you're giving, but you're giving one of your mom's chromosomes, one of your, it's a mixture of chromosomes. Somebody said this as an analogy, and I thought it was a really good analogy, but it's sort of like, when your parents made you, okay, they gave you their deck of cards, and think of like the chromosomes as being a deck of cards. So maybe mom gave you like a red deck of cards, and then your dad gave you the blue deck of cards. And so now you are a mixture of those two deck of cards. And then when you have kids, or when you make sperm and egg, you're kind of mixing up the decks of cards and you're giving them to your offspring. Does that kind of make sense? I hope so. I thought that was a really good analogy. That you're receiving two decks of cards, and then you mix it up, and then you give it out to your kids. So this is meiosis. There's a lot more phases here, but it's pretty much, it follows mitosis, because you've got interphase, prophase, metaphase, anaphase, telophase, and then, telophase over here, and then you have prophase, metaphase, anaphase, telophase. So the difference here is that there's a few differences, but the one that you can see from this slide is that there's actually two prophases, two metaphases, two anaphases, and two telophases, and there's going to be two cytokinesis events. Okay? So let's talk about that. So in meiosis, there's double division, there's two cytokinesis events. Here, in the starting cell, so this is a germ cell, okay, in the testes or in the ovaries, and they're going to go through the S phase too. They're going to go through the S phase. They have their own little, cell cycle too. And when they duplicate their DNA, this is what they're going to look like. You're going to end up with the chromosomes that are double-stranded. The chromosomes will be double-stranded. So this is single strand, right? I only see a single strand. But now, after the S phase, the DNA has doubled. Now I see two of each. It's a double-stranded chromosome, okay? After the S phase. In the first cell division, in the first stage of meiosis, the homologous pairs separate. The chromosome that you got from your dad and your mom, they go in opposite directions. They go in different cells. And then after that, then the sister chromatids will separate, okay? So there's two cell divisions. In the first cell division, it's the homologous pairs, one from mom, one from dad. They will separate. And then in the second cell division, it's the sister chromatids that separate, okay? But I want to ask you guys a question. At what stage is the cell no longer diploid? Remember, diploid means having two sets. Where here do you guys see the cell only having one set? And when I say set, remember, one from mom, one from dad. Okay, I see diploid here. I see diploid here because one from dad, one from mom. Now, that's where they separate. So you guys, it becomes haploid right after the first division. Right after the first division, that's when it becomes haploid. So when the homologous pairs separate, that's when it becomes haploid. And then the sister chromatids separate. It's still haploid, but it's the sister chromatids that are separating, okay? Okay, and then I'll talk more about like how many cells that you end up with later. So in the first step of meiosis, you need to duplicate the DNA. You're going from single strand to double strand. It's kind of confusing, but each one is still considered a chromosome. So this is a chromosome. This is a chromosome. This is a chromosome. That's a chromosome. That's a chromosome. But they're called single-stranded chromosomes. This is still a chromosome. This is still a chromosome. These are all chromosomes, but they're just called double-stranded chromosomes. So there's still the same number of chromosomes. So like I can count here, 1, 2, 3, 4, 5, and then the sixth one is behind my big head. Okay, 1, 2, there's 6 chromosomes. Oops, I can't write, but that says 6 chromosomes. There's also 6 oh my goodness. There's also 6 chromosomes here, but it just so happens that each chromosome is double-stranded. But when you look at the DNA, the amount of DNA, which cell has more DNA? This one does, okay? So after the S phase, you have double the DNA here. There's double DNA because it's double-stranded chromosomes. Whereas over here, the chromosomes are single-stranded, okay? The reason why I'm pointing this out is because every year I have kids like really confused, like, wait, what? Double, single, okay? So there is a S phase in meiosis, just as there is an S phase in mitosis, where the DNA, the chromosomes will double, the DNA doubles, and then you end up with double-stranded DNA. So next slide, okay? This is just going over like why, why in meiosis do they have to double the DNA? Scientists believe that mitosis came first and then meiosis, sexual reproduction, came later. Because if you look at organisms like bacteria, they're very simple organisms, right? They only reproduce by copying that one chromosome that they have. They don't do any of this cell cycle stuff, okay? Okay, so in meiosis 1, so there's two phases. There's meiosis 1 and then there's meiosis 2. In meiosis 1, it's the homologous pairs that separate. In meiosis 2, it's the sister chromatids that separate. In meiosis 1, or the first division of meiosis, the homologous pairs separate. Okay? But here's something very interesting about meiosis 1. In prophase 1 of meiosis, synapsis occurs. Okay, there's this thing called synapsis. What is synapsis? The homologous pairs, one from mom, right? And then the other one is from dad. It says pad, but it's supposed to say dad. I don't know. I think I was going to write pair, but okay. One from mom, one from dad. Synapsis occurs. See how their chromosome, the arms of the chromosomes are like overlapping? They overlap. So your parents' chromosomes, they get really close, they get really cozy, they get side by side, and they form a tetrad. A tetrad is a type of formation. Okay? So tetrad is a type of formation. Synapsis is the process. Okay? So synapsis occurs where the chromosomes from mom and dad, they form a tetrad, they cross over. Do you guys see there's 2 points of crossover? Because what's going to happen, I think I have a better picture of this coming up, but what's going to happen is that the chromosome from mom and the chromosome from dad, they're literally going to switch segments. They're going to break off the DNA and then they're going to like switch pieces of DNA. So now you're going to end up with like chromosomes that have pieces of dad, pieces of mom chromosome on it. You're like genetically just mixing it up. Okay, it's really cool. It's called crossing over, and we'll go over that later. But I just wanted to bring this up because I can see it here in prophase 1. The homologous pairs will form a tetrad. Okay, and then in metaphase 1, they'll line up in the middle of the plate, just like how in metaphase of mitosis, the chromosomes all line up. But notice here, it's the pairs that line up. Remember I'm going to try to draw mitosis here. But in mitosis, all 46 chromosomes line up. I'm not going to bother drawing all 46. But individually, they all line up in the middle. But in metaphase 1 of meiosis, they line up as pairs. Okay, so that's a difference here between mitosis and meiosis. In metaphase 1 of meiosis, they line up together. It's like they're going to do a dance. They're all paired up with their partner, dance partner, in a line. You guys know what I'm talking about, right? Whereas in mitosis, metaphase of mitosis, they all individually line up. Okay? All right, and then in anaphase 1, the homologous pairs separate, and then at that point, you have haploid number. After anaphase 1, you have a haploid number because the pairs separate. Now you only have one set of chromosomes. I see 2 sets right here. 2 sets, right? They're in pairs. Here, the pairs are separated. This is from mom. That's supposed to say mom. This one's from dad. Okay, they're separated. Okay, and then in meiosis 2, this is the second division. In meiosis 2, I see prophase, and then I see metaphase. In metaphase 1, remember the pairs are separated, so they're individually going to line up. Their partners are now gone. And then in anaphase in anaphase 2. Did I say metaphase 1? I meant to say metaphase 2. And then in anaphase 2, the sister chromatids are going to separate. Those sister chromatids are going to go in opposite directions, okay, in anaphase 2. And then telophase 2 occurs, and then you're going to end up with 4 cells. Do you guys see the 4? Do you guys see how we got the 4 cells? So let me go ahead and circle. Like this is, those 2 sister chromatids are in 1 cell. These 2 sister chromatids are in another cell. These sister chromatids are in 1 cell, and then these sister chromatids are in another cell. So I have 1, 2, 3, 4. So at the end of meiosis, you're going to end up with 4 cells, and each of them have half the number of chromosomes because the homologous pair is separated, and each of them are going to have a sister chromatid. Some of you, I can already tell, some of you are just like, what? I don't get it. But you know what, I think I'm going to have more graphics coming up, okay? That will be a little bit more helpful, okay? So what does this look like? This looks a lot like mitosis. I hope I have more. I can always go back and like clarify stuff, okay? So there's 2 stages of meiosis. There's meiosis 1, meiosis 2. With meiosis 1, the homologous pair separates. You're going from diploid to haploid. And then in the second division, it's the sister chromatids that separate, okay? So maybe I'll just draw it here. So you got the homologous pairs, right? 1 from mom, 1 from dad. They separate, okay? So at the beginning of meiosis 2 then, you know, you have, this is the one from dad, this is the one from mom. They're in 2 different cells. That's why they're haploid, because they have 1 set in these cells. They are going to split up. Those sister chromatids are going to split up. So maybe this graphic is, maybe what I'm drawing is a little bit making it easier for you, okay? So the sister chromatids split up. That's how you end up with 4 cells, okay? Because in the first division, you end up with 2 cells, right? These 2 right here, right? It's the homologous pairs that separate. And then in the second division, it's the sister chromatids that separate, okay? But at this stage right here, this is where it becomes haploid because the pairs, the 2 sets separate, okay? And here's just some really nice, actually they're pretty old. These photos look like they were taken back in the 1960s or something, of the different phases of meiosis. But it might be interesting to look at. Okay, crossing over. I did talk about this a little bit. This happens in prophase 1. Okay, so it's important to remember that. Like when they're not lined up yet, but you know what? They pair up. They get together. They get nice and cozy. The chromosomes will overlap. So that's what's going on. And down here in this picture, I hope you guys can see it in the screen capture. But the arms of the chromosomes, they overlap. This is, so synapsis is a process, right, of them overlapping or crossing over. Sometimes it's just called crossing over. All right. They form a tetrad. You know what? The reason why it's called a tetrad, because tetrad means 4, right? How many chromosome arms do you see? I see 4. 1, 2, 3, 4. How many sister chromatids do I see? 1, 2, 3, 4. Okay. And then here, do you guys see, excuse me, the chromosome from your mom, the maternal chromosome, now has pieces of the paternal chromosome from dad. The dad's chromosome now has maternal chromosome or DNA, maternal DNA, okay? So what we're doing here is we are producing amazing, unique, you're basically mixing up your genetics that have never existed before. Like this combination of chromosome, a mix of your mom and dad has never occurred before on this planet. So when somebody makes gametes, there's no 2 gametes that are the same, because actually this crossing over is very random. I didn't mention that earlier, but you know, in these PowerPoints with the drawings, it always happens at the tips, okay? But it doesn't have to happen there. It can happen anywhere. It can happen anywhere along the chromosomes. Like you could actually, and I've seen pictures of like crossing over where the dads or the moms are completely like crossed over, where you can't even see the chromosome underneath it because they're like so cozy and they're crossing over at many different points. You might end up with sister chromatids with 5-10 places where they've crossed over. Okay, so crossing over is it's an amazing process because you are mixing up your parents' genetics that have, and you're producing combinations that have never ever existed before. So when you think about it, no two eggs are the same, no two sperm are the same. That's why when you have a big family, like lots of kids, no two kids are exactly the same. Your brother, your sister, you guys are unique, genetically unique because of crossing over. Okay? Even when I have two sisters, all of us, all three of us are so different from each other. We look different, we act different, personality, everything is so different about us because of crossing over, right? And it's a random process too. You don't know which gamete's going to get that gene. It's just this random mixing over. So just think about it. You are very unique because the gametes that made you, the egg from your mom, the sperm from your dad, was genetically unique to make you, which is a completely unique person. So I hope you feel better about yourself, okay? So here's just another diagram just showing you, right, crossing over, and you're producing new combinations of chromosomes, okay? What are the advantages of crossing over in sexual reproduction? I mean, you're going to get genetically unique gametes, and you want that in a population. When we go over evolution, population genetics, evolution, you'll see that the more genetic diversity there is, the safer the population is in terms of survival. You don't want everyone to be genetically the same. Or else if there's some kind of virus, some kind of pathogen that can kill one, then it can kill many of the population. You want to mix up the genes. And you do see this with breeds of dogs. They say that, mutts, dogs that have a lot of different kinds of breeds in them, they're a lot healthier, they live longer. You don't see a lot of the genetic mutations that you see in purebreds. I'm sure you guys have heard of that before. Okay, so here's a graphic showing you comparison mitosis versus meiosis. Okay, with prophase of mitosis, the chromosome, you start to see the chromosomes, right? In prophase of meiosis, the thing is here is you start to see the tetrad forming. Okay, remember the homologous pairs, they get together, they get cozy. I don't know why I keep on saying cozy. Is that annoying? Well, anyways, the crossing over takes place. And then also in metaphase of mitosis, each individual chromosome lines up at the metaphase plate. So imagine for us, all 46 individual ones will line up. But in meiosis, metaphase 1, it's the 23 pairs that line up. Okay, so 46 individual ones line up in mitosis of metaphase, but it's the pairs that line up in metaphase 1 of meiosis. And then at this point, when the pairs split up, that's when it becomes haploid. Okay, in mitosis, it's always haploid, right? You're always, because we're making identical cells to the parent cell. And this is just a picture showing you after the S phase. So after the S phase, you see the double-stranded chromosomes. But you end up with, identical cells to the parent cell, okay? And then they'll go through the S phase, and then you'd see the double-stranded DNA. Okay, and then after meiosis 1, then you end up with haploid cells. Then the sister chromatids will separate. And then you end up with 4 cells with genetically unique combinations of paternal-maternal genes. Okay? So let's compare 1 cell division versus 2. Mitosis, daughter cells are genetically identical to the parent cell, whereas in meiosis, the cells are genetically unique or different from the parent cell. In mitosis, we're only making 2 cells. In meiosis, we're ending up with 4. We're going from diploid to diploid, whereas meiosis is diploid to haploid. We're producing cells for the purpose of growth and repair and development in mitosis. And what is the purpose of meiosis, everybody? For sexual reproduction, to produce gametes, okay? And then also in mitosis, there's no crossing over, whereas in meiosis, there is crossing over. So let's put it all together, right? In an individual, right? In a sexually mature organism, you have 40 adult, you have, so maybe I'll just write adult here, okay? You have 46 chromosomes. Oh, I should say these are the germ cells, right? Those are the germ cells that are going through meiosis in the adult. In the ovaries, ovary, and then testes. So in meiosis, they're producing gametes with half the number of chromosomes. And then at fertilization, you have 23 plus 23, which makes, 46, a zygote. And then that zygote is going to go through mitosis and develop into a little cute little baby. Okay, and then I always have students ask me, what the heck is that on the right side? That's the placenta that is embedded into into the lining of the uterus. Okay, so this is the uterus, this is the placenta. See how the blood vessels are in red and blue? The blood vessels between the little fetus and the mom they're intertwined so that she can provide oxygen and food for her growing baby. Okay? So let's talk about sexual reproduction. It is, it's an advantage, okay? So like speaking from an evolutionary genetic, population genetics viewpoint, there's an advantage to reproducing sexually because number one, you're gonna get genetic variation, okay? And you can get genetic variation because of several reasons. And we've already talked about this one, okay? We've already talked about crossing over. I mean, because of sexual reproduction, you're producing gametes by meiosis, which includes crossing over. You're mixing allele. Oh, we haven't talked about alleles yet. But alleles are the different genes for a trait, like brown, green, green, okay? Basically, alleles are the different genes for a trait, like you're mixing it up, okay? Okay, so let's first talk about this one. We already talked about crossing over, okay? But we get genetic variation by genetic recombination, and I am going to talk about that by looking at the drawing at the bottom, and I hope you guys can see it. Okay, it's called genetic recombination or independent assortment of chromosomes, random alignment of homologous chromosomes in metaphase 1. Okay, so remember how I said, and maybe I'll try to draw it. No, I like this drawing at the bottom. I hope you guys can see it. So you know in metaphase 1 how the homologous pairs, they pair up. They line up at the metaphase plate. And then the way that they line up in the metaphase plate, the orientation of like where the maternal chromosome is, where the paternal chromosome is, it's completely random, okay? Like in this this situation, number 1, so there's 4 examples here of 4 different possibilities. There's more than that, okay? Because we have 46 pairs. I'm sorry, 23 pairs, okay? But in this picture, there's actually 3 pairs of chromosomes, but we have 23 pairs. So the independent assortment, the amount of different kinds of ways in which the pairs line up is, I think it's, I'm going to go over this on the next slide. I think it's 2 to the 23rd power, the different variations, okay? Anyways, we have, let's say that the chromosome from dad is on the left side, chromosome from mom is on the right side, and it just so happens that all the ones from your mom is on the right side, all the ones from your dad are on the left side. And then there's also option number 2. You have dad, dad, and then mom at the bottom. And then you have mom, mom, dad at the bottom, okay? In this one, it's dad, mom, mom. mom, dad, dad. So do you guys see, like, you can have like different variations of how the paternal and maternal chromosomes, they line up in the metaphase plate. Then you're going to end up with so many different kinds of gametes. I wish I had a better picture so that, or like a whiteboard. I wonder if, and then also the thing about this PowerPoint pen is I can't change the color. I would like to change the color so I can have like maternal, paternal examples. Let me see. Yeah, it doesn't. Oh, pen color. Okay, let's do it. I'm gonna write it over here then. I'm gonna draw it over here. So, if I have from mom here, yeah, from mom here, and then I'm gonna draw this one over here. And thank you guys for, I can already tell you guys are probably like, oh, this is taking too long. So please be patient with me. So this is situation number 1. Okay, so this is a cell. This is situation number 1. And then this is scenario number 2. Okay, so see how, do you guys see the difference between the left side versus the right side, right? If they line up like this, where all the maternal ones are on the left side and paternal ones on the right side, you're going to end up with four gametes like this, okay? Remember the homologous pairs separate, and then the sister chromatids separate, okay? You're going to end up with, now I got to get the red one out. Oh, let's do the blue ones first. You're going to end up with gametes, 2 of them with the, and then they go this way. Yeah, they go that way. Okay. Yeah, and then 2 of them with the blue sister chromatid, and then where's red? Hello, there it is. And then you're going to end up, oh wait, you know what? I forgot. It's this. That's why I was confused. It's like this. Oh, that looks horrible. I apologize. I was going to say, I'm confused here, but it's because I forgot to do this. Okay? So you're going to end up with 4 gametes, red, 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 blue, 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 blue in this scenario, okay? But if you take a look at the this scenario on the right, so let me just draw a line here. You're going to end up with 4 gametes with different combinations, okay? Like this one's going to have a red, blue, and then let me grab my red. I wish I could draw it all out here, but there's not a whole lot of space. So this is what you're going to end up for this situation, okay? So this is what we call independent assortment, because the way that the chromosomes line up together is independent of each other. Like the ones on the top, they don't influence the ones on the bottom below them. So you could have red, blue, or you could have red, blue, blue, red underneath them. And we have 23 pairs. All 23 pairs line up together. And then the one, the way that the maternal, paternals, they line up, the orientation is completely random. See how I ended up with 4 different kinds of gametes on either side? It's incredible, the different amount of variations of gametes that we can end up with, okay? Oh yeah, and then here's that number, 2 to the 23rd power. That's 8,388,608 different combination of gametes, just based on independent assortment alone. The way the 23 pairs line up, the way they're distributed to the gametes. And then crossing over is completely random, as I was trying to say to you guys. There's it's random, so there's really an infinite variety of gametes that can be made just from crossing over alone. And then also, fertilization is completely random. So you know how I said that, females have about 200,000 eggs when they are sexually mature? So it's that 1 out of 200,000 eggs that is released a month. So 1 out of that is going to be released. So think about it. You you are a product of that one, just by pure chance, that one egg being released that month. And then for your dad, there's hundreds of millions of sperm, and that one egg and that one sperm out of the hundreds of millions of sperm that day or that moment or whatever you guys want to call it, fertilized. They came together to make you a genetically unique individual. Okay, so any two parents will produce a zygote with 70 trillion possible diploid combinations, not even counting crossing over. Okay. By the way, this is from our very old, I think it was like a 2006 AP bio book. Here's couple number 1, here's couple number 2, and the question is like there's like 4, each couple had a boy and a girl. So the question is like, out of the 2 girls, which girl belongs to which couple? Which boy belongs to which couple? And then maybe you guys can take a second to guess. Okay, well, I'll tell you guys. So this girl and this boy are siblings. I think a lot of people could have guessed that. They look similar, but different, right? And they are the children of couple number 1. And then the 2 kids in the middle are the children of couple number 2. Okay? I just thought that was really cute. So as I was saying about like siblings, they look, you guys, if you have siblings, you look very similar, but different, right? Because no 2 sperm are the same. No 2 eggs are the same. To me, it's just this, it's such an amazing thought to me. Like when I think about my son, I absolutely adore my son, you know, but if it was a different egg, if it was a different sperm, it wouldn't be him, it would be another kid. If it was a boy, he would have the same name, because I always wanted a boy named Noah. So he would be still named Noah, but he would be a different kid with a different personality. You know, similar, but different, right? Because it's his hypothetical brother, right? Similar, a sibling, but different, okay? So here's some pictures of famous siblings. You have the Hemsworth brothers. Is it them 3 only, or is there 4? I don't remember. I think it's them 3. And then the Olsen sisters at the bottom. When I was growing up, the 2 oldest Olsen sisters, which are the petite ones, they were on a show called Full House. But you guys all recognize, that's Elizabeth Olsen, right? She plays in the Marvel films. Who does she play? Oh, there's that new show, WandaVision, right? Wanda, she's Wanda, right? I cannot believe I'm... Anyways, whatever. And then you have the Jonas Brothers. Oh, wait, that guy, he's not a Jonas Brother, but there you have the Jonas Brothers, okay? So as I was saying, sperm production happens in the testes by meiosis. As I was saying earlier about, like, out of 1 in hundreds of thousands of sperm, there's about 100 to 600 million sperm in an ejaculate. So that's like a lot of sperm. And the fact that that one sperm reached the egg to make you, to me, is just amazing. Okay. Okay. And then I did mention this earlier about how in women, we're born with about 2 million eggs, and then by the time we reach sexual maturity, there's only about 200,000. The numbers are varying. Like some websites will say 100,000 to 200,000. So I'm just going to go with 200,000 that are in the ovaries. But meiosis actually doesn't occur, or doesn't finish, I should say, doesn't finish until fertilization. Until the sperm enters the egg, it doesn't actually finish the whole process of meiosis. Okay. But one very unique thing about egg production, and egg production is called oogenesis, and spermatogenesis is the name for sperm production. Okay, oogenesis. Every time I say that word, it's like a song. Oogenesis. Okay. Eggs in the ovaries are halted before anaphase 1. So actually, when females are born, we have those millions of eggs. They are in anaphase 1, and then meiosis 1 is completed during sexual maturity. you know, when she goes through ovulation, then it completes it completes meiosis. What is it called? Meiosis 2. Okay, is it meiosis 2? Yeah, it shows it right there. So here's what's going on. So here's the primary oocyte. This is the cell that's going to go through meiosis. Remember, the homologous pairs, they separate. Homologous pairs are going to separate. But here's the thing. It's unequal division, unequal division. One cell is going to be super fat. The other cell is going to be super small. And that super small cell is going to die. Okay? The fat one's going to live on. This big juicy egg is going to live on. Because remember, eggs have to provide all the nutrients for that little zygote. You know, whenever I draw like an egg and then the sperm is really small. It doesn't carry much to the egg except for DNA. Okay, most of all the nutrients come from the egg, and then the mitochondria and all the organelles. The organelles are usually provided by the egg. I've heard of some stories where some of the mitochondria comes from the dad, but for the most part, mitochondria comes from the mom. Okay, so that's called mtDNA, which is mitochondrial DNA, is usually from the mitochondria from the mom, from the egg. Okay, so after the second division, at ovulation, the second division happens. And again, it's unequal cell division. You have one juicy egg and then the other polar body, the dinky little tiny cell, it dies. Okay, so the polar bodies are the cells that die, and then the other cells that are nice and big and juicy, those are going to become the egg. That sounds kind of gross, juicy egg, huh? And again, the reason why is because we want to create a big egg with all the nutrients that are going to help that zygote survive. We do not want to create 4 small cells like in sperm formation. In sperm formation, so let me go back. In sperm formation, you are producing 4 small cells. 1, 2, 3, 4. Okay? But in egg formation, you're producing one fat, juicy egg. That's going to have all of the nutrients. Okay? Gosh, I just realized like I shouldn't be saying juicy. That sounds a little bit weird. And then also like on the AP exam, please don't say big juicy egg. Just say big volume egg with all the nutrients it needs, okay? All right, let's see here. We're not going to go into all the details, but you know, the ovaries have those hundreds of thousands of eggs. Every month, one follicle develops, okay? And then the follicle erupts, releasing the egg, and then there are all these little cilia that cause the egg to be swept up into the fallopian tube. And then you guys can see in this picture, that's where fertilization takes place, in the fallopian tube. And then it starts to develop, sorry, it starts to go through mitosis and develop and end up in the uterus. And then it will implant in the uterus like at around day 10, okay? All right, I'm gonna have to pause the recording here and I'll be back. I mean, I just realized I only have like one more slide left, so why did I just stop? Oh, you know what, it's because right now it's like 2:13 and I have office hours in 2 minutes, so I'm gonna try to rush here. You know, there's different life cycles found in the animal kingdom. Okay, you don't need to know too much of this, okay? But the life cycle of the human that we went over is the one that you need to focus on. Okay, so I think that that's it. And then you know these questions at the end, we'll go over those in class. Okay, or if I have time today after office hours, I might come back and I might record me going over this. I think that you guys probably would appreciate that, huh? You know, have you heard of the Baldwin brothers, right? They're siblings and they all look very, oh yeah, you guys have definitely heard of Alec Baldwin, right? And then one of these guys is the dad to that girl who's married to Justin Bieber, I forget her name. Eggs are precious, sperm is cheap. And the reason why it says this here is because, you know, we'll, you know, I have to go to office hours, but I'm going to talk about that. It's very interesting. You guys will hear some stories when you go to college about selling eggs, and they are You can get a lot of money for selling eggs, okay? Whereas sperm, you won't get that much money for selling your sperm. Okay, I will be back. Okay, so as I was, all right, so as I was saying, oh wait, I'm also in a Zoom, so that's why that popped up there. But I am muted, so okay. As I was saying about eggs are precious, sperm is cheap. What does that mean? When I was in college, which was decades ago, I would see these really weird advertisements in the school newspaper. I went to USC, and I would see these weird advertisements. It would say $200,000 for eggs. I was like, whoa, what does that mean? So when I read it carefully, there were these rich couples that were infertile, so they couldn't have their own kids, and they were willing to pay a young college student money, hundreds of $1,000s, and so for her eggs. In order to get the eggs, though, it's pretty complicated. You have to go through hormone therapy, like you have to get daily injections of many different kinds of hormones to prepare the body to just release a bunch of eggs at the same time. And then you have to have surgery to go get the eggs out of the ovaries. It's a big deal. So that's why at that time it was really expensive, because part of the technology when I was going to college in the early mid 90s, I don't remember. The technology was not that great, okay? But now that things have improved, it's a lot easier to get eggs. So I just looked online. You can sell your eggs if you meet the criteria. You can sell your eggs. I think it was like $8,000 to $10,000 per cycle, okay? And a cycle could take like 6 to 8 weeks. So you could get $10,000. But then you're selling your eggs to a couple that is infertile, that can't have kids of their own. Maybe for some reason, the female partner, can't produce eggs on her own, something like that. Or maybe you have a couple that just can't biologically have kids on their own, so they need an egg donor. So that's what I mean by eggs are precious. And sperm, you can sell your sperm, I think $50 to $70 per donation. And as you guys know, like with egg donation, it's like 6 to 8 weeks it takes, but for sperm donation, it's just whenever you want, right? So I think I read online, like somebody that is a healthy male can make up to $1,000 a month, which just from donating sperm. But here's the thing, like, are you willing to be the father of many anonymous children? I don't know. Okay. All right, what's Fillmore asking here? What are the disadvantages of sexual reproduction? And we talked about that at the very beginning. One of the disadvantages of sexual reproduction is that there's so much energy spent into producing gametes, right? Meiosis. But if you were to reproduce asexually, you don't have to spend a lot of time and energy doing that, right? And then also with sexual reproduction, you have to find a mate. Maybe think of yourself as being a lonely mountain lion who's miles and miles away from the next, mountain lion potential mate. It's a lot of work. Okay, a lot of energy goes into sexual reproduction. All right, so let's look at some practice problems. And you know what? I have to say, a lot of the questions at the end here, they're very similar to each other. It's kind of repetitive, okay? And I think, excuse me, these questions came from like a practice exam, and they just all have the same type of questions, okay? How do cells, but I put them in here because they actually make you think about the DNA, the amount of DNA in meiosis. How do cells at the completion of meiosis compare with cells that have replicated their DNA and are just about to begin meiosis? Okay, so maybe just to help us out, maybe I can go ahead and draw like on top of here, you're gonna have, it says replicated their DNA. So all the chromosomes are double-stranded, okay? And we have 46 of them, right? We have 46 of these double-stranded. So altogether, how many pieces of DNA do we have? We have altogether 92 chromatids or 92 DNA pieces, right? Chromatids. And then at the end of meiosis, right, you're going to end up with 1, 2, 3, 4 cells. Each of them, I'm not going to draw all 46, or actually 23, right, because it's half the number of chromosomes. I'm not going to draw all 23 of the sister chromatids. So let's say, look at the answer choices. They have twice the amount of cytoplasm and half the amount of DNA. Okay, how do the cells at the completion, so at the end, do they have twice the amount of cytoplasm? That kind of doesn't make sense. That means that they're like twice as fat and juicy, right? There I go again with juicy. So I'm going to cross out this answer choice. Okay, B, they have half the number of chromosomes and half the number of DNA. Okay, so what that means is that there's 23 chromosomes and half the amount of DNA. So if you kind of think about it, I mean, there's 92 chromatids, but how many chromatids actually are in each of these? There's 23. 23. So is 23 half of 92? No. I feel like I'm lagging and it's so distracting. I hope that when I actually put up this video, it's not lagging. But anyway, so it's not B. They have the same number of chromosomes. That's not true, right? Because we started off with 46 or 92 chromatids and half the amount of DNA. That's not true. So we can just immediately cross it out. D, they have half the number of chromosomes. That's true. We're going from 46 to 23, okay? And 1/4 of the amount of DNA. So we're having, that means there's half the number of chromosomes, which is 23. And then 1/4, so what's 1/4 of 92? So if we divide 92 by 4, that actually gives us what? 23, okay? So 23. So let's leave this one open. I'm pretty sure that D is the correct answer. Let's take a look at answer choice E. They have half the amount of cytoplasm and twice the amount of DNA. That makes no sense. We're starting off with 92 chromatids, the amount of DNA, and it's saying twice the amount of DNA? No. Okay, so the answer is D. Okay, and this is kind of related to the previous question, but in graphical form, sort of. What number represents G2? So you guys remember the cell cycle? So we're still talking about meiosis. How in meiosis, those starting cells also go through the cell cycle, right? So here's the, this is interphase, right? And then this is maybe, no, I was going to use a different color, but forget it. G1, S phase, G2. Remember, S phase is where it's doubling the amount of DNA. So we're going from 46 of these guys, and at the end of the S phase, we have 46 of these guys, right? So we're doubled the amount of DNA because we have DNA synthesis happening. DNA is replicated. We have this chromatid has replicated itself. Now you have sister chromatids. So which number represents G2? So we still have 46 of those guys at that point, right? It's not until mitosis, or in this case meiosis, it's not until meiosis where you start to see changes in the chromosome number. So which number represents G2? DNA content in the cell. 1 time, 2 times, 3 times, 4 times. So how many more? So 46 is 92 chromatids, right? Chromatids. So how much DNA content is in G2? Okay, there's phase 1, phase 2, phase 3, phase 4, phase 5. Okay, so if you take a look here, this is 2x, which can stand for the homologous pairs, like 2 pairs. So let's say this is actually like 2n, right? 2n. And so when the DNA duplicates, you're going to have 2n, but then it's all double-stranded. And so you're going to have 4 times the amount, double the amount of DNA, which is 4 times, okay? So the answer is going to be 2. You might be confused right now, but that's okay. So if we have because we're going to go over the rest, okay? So if we have, at this point, 46 of these guys, remember during meiosis, the homologous pairs they have to separate, okay? So then at this point, we're going to have like the homologous pairs, they're separating, okay? So now we're back down to, this amount of DNA because you have sister chromatids. But then remember when the sister chromatids are separating, that's this thing that's happening right here. The sister chromatids are separating. So maybe I should draw it like they're separating. And then at point 5 or V, you end up with a sister chromatid. Okay? Maybe I should draw like, oh, yeah, okay. So I'll just draw it like this. But you have homologous pairs, okay? So homologous pairs, then the homologous pair, maybe I should have drew it like this from the beginning. Sorry, guys. And then, yeah, maybe I'll draw it like this, where it's going like this. Yeah, this makes more sense. So you have the homologous pairs, single-stranded, 2N. Remember, 2N means 2 sets. And then in the S phase, the chromatids or the chromosomes will duplicate. They'll duplicate. So you have this amount of DNA. And then the homologous pairs are separating in this process. You're back to having this amount of DNA, which is the 2 sister chromatids, which is equal to the single-stranded 2. Okay, very confusing. And then when the sister chromatids separate, then you're down to a chromatid. Okay, so you're going to have in each cell, so when you think about, so I got to draw it like this. In each cell, you're going to end up with a single strand. Okay, one single strand, so 1/4. I hope that, this one's a tougher one. I think this one's a tougher one. You really have to like visualize, or see, maybe you can have in front of you a diagram of the entire meiosis process. Okay. Oh, this graph again. Which number represents the DNA content of a sperm cell? You guys, same thing, right? Which one is a sperm cell here? At the end of meiosis, it's going to be this guy, right? At the end. So it's going to be 5. So E is the correct answer. Because it's going to have the least amount of DNA. It's just going to have that one single-stranded sister chromatid. Which of the following would not be considered a haploid cell? So what's not haploid? A daughter cell after meiosis 2. Remember by the time of meiosis, at the end of meiosis 1, when the homologous pairs separate, it's already haploid. So it can't be this because it's saying not haploid. It's at that point already haploid. What about gametes? Gametes are haploid, so it can't be that one because it's asking not haploid. Okay, so not haploid. Sorry, I'm a kind of a visual person. Daughter cells after mitosis and gametophyte generation of a plant. You know what, guys? We're not going to go over the gametophyte generation of plants. But basically, a gametophyte is a haploid, multicellular life cycle of a plant. Okay? Don't worry about it. Our students had to know that back like 10 years ago, but it's been too long. That's maybe how old this question is. Okay. Cell in prophase 1. What do you guys think? Remember, so in meiosis 1 and meiosis 2, prophase 1 is the first of meiosis 1. At that point, it is still diploid, so not haploid. So I'm going to circle D, but let's take a look at E. Cell in prophase 2. By that point, it's already haploid. Okay? So maybe I'll draw it just really quick just to clarify. Okay? So here's a homologous pair starting in meiosis 1, okay? So this is, let's say this is metaphase 1. They're lined up, they're, side by side. And then at the end of meiosis 1, anaphase 1, telophase 1, the pairs are now separated. Now you have just one set of chromosomes. Remember, 2N refers to one from mom, one from dad. But at this point, after telophase 1, the homologous pairs are separated. Now you only have one pair, 1N. or haploid. And it still stays haploid because you still have one set. I don't know if you guys can see what's on the bottom of this page, but each gamete has a single chromatid. Okay? So it's still haploid down here. Haploid comes into play once the homologous pairs, they separate. Okay, I think this is the last question. A cell in, this is very similar to the first and second question. A cell in G2 before meiosis compared with, I can't see it because my big face is in the way. Let me move this. A cell in G2 before meiosis compared with one of the 4 cells produced by that, okay, meiotic division has, so it's asking compare G2 to those that is produced by meiosis, basically the sperm or the egg, okay, the gametes. So it's asking you to compare the cell in G2 with the one in gametes. And so remember, it's gone through the S phase, now it's in the G2, and at that point, we have 92 pieces of DNA because we have 46 of these guys. Oh, is this confusing, you guys? Okay, we have 46 of these guys, and if you count each sister chromatid, that's 92 chromatids. That's 92 pieces of DNA. I hope you guys got that. I hope you understand that. I could always clarify during class, okay? So we're comparing that to a gamete, which is just going to have 23 of these guys. Okay? So let's see. A cell in G2, blah, blah, blah. Twice as much DNA and twice as many chromosomes? Does 92, is that twice of this? 23? No. Okay. Let's look at B. 4 times as much DNA. That's possibly the answer. And twice as many chromosomes. That sounds correct, doesn't it? Because it has 92 pieces of DNA versus 23. But then, it has 4 times as much DNA because it has 92 pieces of DNA versus 23 pieces of DNA. But over here, we have 46 chromosomes and 23 chromosomes. I think maybe some of you guys, I might be confusing some of you because of the 92, 46, 32, 23 thing. So keep in mind that we have, at the beginning of meiosis, we have 46. Okay, we have 46 of these double-stranded chromosomes. Double-stranded chromosomes. So 46 of the double-stranded. I'm just going to write DS. Okay? And then each one is considered a DNA molecule. Okay? So then that's why I got 92 pieces of DNA. Because there's, each one has 2 pieces of DNA. Each one of the 46. So double 46 is 92. Now I'm starting to confuse myself, but I think that makes sense, okay? All right, let's take a look at answer choice C, just in case. 4 times as much DNA and 4 times as many chromosomes? No. Half as much DNA? No, G2 does not have half as much DNA, but the same amount of chromosomes? That doesn't make any sense. Half as much DNA and half as many chromosomes? No. Okay. So the answer is D, B, answer is D. 4 times as much DNA, because there's 92 at G2, and twice as many chromosomes, 46 versus 23. Okay? I hope that makes sense. Again, I can clarify more during class, okay? So that's chapter 13. It wasn't too bad, I hope. It's actually kind of interesting, right? Sexual reproduction, meiosis, production of gametes. I think, in my opinion, it's pretty interesting, and I hope that this video was helpful and we can go over any questions that you might have in class. All right, thank you. Bye-bye.